there are 6 different possibilities to block the neurotransmitter, the neuromuscular transmission. And I told you that there are drugs that we use in medicine by which we want to block this neuromuscular transmission. There are diseases by which the neuromuscular transmission is affected and results in muscle weakness. Those two examples I brought to you, autoimmune diseases which builds antibodies against acetylcholine receptors, which is myasthenia gravis, and result in muscle weakness, and another one, Lambert-Eaton syndrome, by which antibodies are produced against the calcium channels, okay? And also we have seen that many animals or bacterial toxins can block this neurotransmission. As you see, for instance, botulinum toxin affects here to cleave the proteins of the core complex, and therefore the fusion of the vesicle with perisynaptic membrane is inhibited. So, but 2 of these groups are important for medicine, for you as physician, for treatment. And that is mostly here group 4 and also group 6. By 4, you see here are 2 different substances. One is the agonist for acetylcholine. For example, where is plastia? An example for that is succinylcholine, for example. That's a drug which anesthesiologists use to relax the muscle during operation. What this drug do is that its structure is very similar to acetylcholine. If you apply it, then the drug comes here and binds with the acetylcholine receptors, like acetylcholine, but and open also the receptor. So it is like normal acetylcholine. The difference is that acetylcholine, as you have learned yesterday, is continually degraded or metabolized by an enzyme that you have here, and it is called acetylcholine esterase. After it sits on the receptor, the enzyme destroys it, and then the next molecule must sit there. And this succinylcholine is not metabolized by this enzyme. Therefore, it can sit there on the receptor, open the channel, and as long as it is there, okay? And it means that the inlet potential which is produced is a prolonged inlet potential. It is not like by acetylcholine to produce inlet potential and this goes away, but it takes longer, it stays there. You say it is a permanent depolarization of this membrane. And then you will argue, if I depolarize this membrane, then I must reach the threshold of voltage-gated sodium channel and elicit an action potential. So it will produce an action potential, and that is not the paralysis of the muscle. And that is true. At the beginning, as you apply it, it produces repetitive muscle contractions. So a series of muscle contractions. But then by the time, after a short while, it leads to muscle relaxation. And the question is, where does the muscle relaxation come? And the answer you know already. You have learned in the foundation block that in order to produce an action potential, a second action potential, you must the membrane must repolarize back to normal, because this repolarization brings the sodium channels to the point that they can be activated again. Otherwise, if you don't repolarize the membrane, then the sodium channels are in inactivated condition. In an inactivated condition, you cannot produce a second action potential by any stimulus, even if the stimulus is very strong. And you learn that phase of action potential as absolute refractory period. And now this permanent depolarization doesn't allow the membrane to come back, to repolarize back to normal. It means all sodium channels remains inactivated. If they are inactivated, then any depolarization, even if that is larger, cannot produce a second action potential. And there is where you get a paralysis of the muscle. You cannot produce more action potential, and therefore the muscle cannot contract. So this permanent depolarization causes also a neuromuscular, let's say, signal transmission, because it doesn't allow to produce a new action potential. The other group which physicians use is the antagonist of acetylcholine. For example, this tubercularity. If you apply this drug, administer this drug, it goes and sits on the receptors of acetylcholine. And here the difference is it sits on the receptor like acetylcholine, but does not open the channel. The channel remains closed, and therefore sodium cannot move through these channels to produce end-plate potential. Okay? And therefore the neuromuscular transmission is blocked. And these are antagonists or agonists of acetylcholine for neuromuscular transmission block. Another group is this one, group 6, acetylcholine esterase. If you give inhibitors to these enzymes, inhibitors of that enzyme, for instance, fusiostigmine, then this enzyme cannot degrade or cannot metabolize acetylcholine, cannot break down acetylcholine. And now this acetylcholine, which is released here, accumulate in the synaptic cleft because it is not metabolized, it is not break down. And then you have a lot of acetylcholine which sit on these receptors and produce a permanent depolarization. Then by acetylcholine itself, you have effects like succinylcholine, like acetylcholine agonist, a permanent depolarization which inhibits the creation of action potential, and therefore you get a paralysis of the muscle. Got it? Although these groups of drugs are used in anesthesiology to relax the muscle during operations. So now I move to lecture of today. Yes. I'm sorry, I cannot hear you. That is really a very difficult question for me. Do I need to learn these or not these? As I was a student, I wanted to know everything which was available to me. I would say what is written in the text below my slides, they are the main point that you have to know at all time. The text, which is my slides, are always given a description below the slides. Those are the main points. The more you learn, the better it is for you. I can never answer you don't learn it, because that is a behavior against academic life. I'm still every day I'm learning something. Okay? It depends on your time, and what you know. But the main important things are described below the slides. Okay. Now I go to the next lecture. Okay. How can I go to a different mode here? It is why it shows now different. Hello? Oh, that is PDF? Oh yeah, thank you. You see, if you become older, you get problems. No. So this slides now we should tell you what you have to know from histology, because I don't talk about histology so much. Now we want to start with muscle contraction. These are the learning objectives that you read by your own, and you see that all of them will be covered. They will be covered. And in the muscle contraction, in the process of muscle function, there are three processes which are connected to each other. The first process is a muscle action potential. And we have learned how the muscle action potential is produced. It must come from the nerve to the muscle, transmitted to the muscle. That is the first step. And then following and that is electrical process. Following this electrical process, there will be a mechanical process. It means the actin and myosin filaments are sliding against each other. And that produces the mechanical power. We have a mechanical process following the electrical. And then both these processes, electrical and mechanical, are connected to each other with calcium ions. Okay, calcium ions connect these two. Therefore, the name of it, we call it electromechanical coupling. It means electrical processes coupled with the mechanical process through calcium. It is an electromechanical coupling. Or another name, excitation contraction or excitation contraction coupling. Excitation relate to electrical to action potential, and contraction is the mechanical process. You see, by physiology, most of the things you know if you really look at the name. The name tells you a lot what is happening there. Okay, now we learn the action potential, that the muscle itself cannot produce an action potential. The action potential is transmitted from a nerve to the muscle, and now after it is produced, then it's propagated over entire membrane of the skeletal muscle. And you have learned in histology that the plasma membrane of the muscle cells moves deep into the cell. In those areas you call transverse tubule. Okay? And if you look at the muscles, there is a muscle cell, and then here, and the transverse tubule goes deep into the muscle, and on both sides of this transverse tubule, you have in the cytoplasm the endoplasmic reticulum. Very close, the cisterna of endoplasmic reticulum come very close to the membrane of transverse tubule. And that is what is shown here. You see here is the transverse tubule, and that is the inside of the cell, and that is also the outside, the inside of the cell. And you have the Zarcoplasmic Reticulum, one here and one there. This here is the lumen of transverse tubule. That is outside of the cell. Here is the outside of the cell. And these two, a little bit red here, that is the inside. And you see this structure, these three together is called, how do you read this? Triad? Triad? Okay, because there are three. 1, 2, 3. So, and that is very important for the release of calcium, this structure. And now we want to see how, roughly in summary form, how the mechanical contraction happens, and then I go step by step forwards, okay? Here you see the muscle cell membrane, and that is the transverse tubule. You see that these membranes go deep into the muscle, and now an action potential is produced here at the plasma membrane, and it propagates along this membrane and comes here in transverse tubule. And now here in the cell, on both sides, you have Zarcoplasmic Reticulum. What happens here that in the membrane of the cell, here transverse tubule, you have receptors which are calcium channels, and they are called DHP receptor, dihydropyridine receptors. And also in the membrane of Zarcoplasmic Reticulum here, you have another receptors. They are also calcium channels, and they are called ryanodine receptors. So in both membranes you have receptors. And this action potential change the conformation of these receptors so that this ryanodine receptor opens and the calcium comes out of the Sarcoplasmic Reticulum into the cytoplasm, and that release of calcium triggers the actin-mucin cross bridges, also the actin-mucin interaction. Yes, please. That they are all written in your text. That is DHP, dihydropyridine, and the other one is ryanodine receptors. Okay, and then after calcium is released, actin and mucin can bind with each other, and the contraction begins. Now we go forward step by step. The first step is action potential, you know. The second is calcium release. Where does the calcium come and will be released? There are two sources for calcium to be released into the cytoplasm of the cell. One source is here out of the cell, that is the cell membrane. Here is inside of the cell, that is outside. That is the concentration of calcium out of the cell. And you have learned that is around 2 millimole per liter. And if you are very exact, it is about 1.8 millimole calcium per liter of blood plasma of extracellular fluid, or in all solution outside the cell. And if you go into the cell, then in the cytoplasm of the cell, you have a calcium concentration of 10 in the power of minus 4 millimole per liter. It is 10,000 times less than the outside. Okay? This value is 10,000 times less than this one. So, inside calcium concentration is extremely low. If you, even it is that low that probably each cell has 1 or 2 calcium free there. It's very low. And then you have sarcoplasmic reticulum in the cell, which has a very high calcium concentration. You see it is double as much as outside. It is almost, again, 10 times higher than in the cytoplasm of the cell. Therefore, if you want to increase the calcium concentration in the cell, in the cytoplasm, then you have two possibilities. Either you bring calcium from the outside or from the inside of the sarcoplasmic reticulum. Okay? From inside, from sarcoplasmic reticulum, or from outside. And now we have seen in this picture, better you can see that here is the tubule, the transverse, that is the inside of the cell. Here is the lumen of the tubule, this transverse tubule, and that is the membrane of the sarcoplasmic reticulum. It means that what you see here is inside of the sarcoplasmic reticulum. Here is the lumen of the tubule, and as I told you before, in the membrane of the tubule, you have DHP receptor, dehydropyridine receptor. They are L-type calcium channel. They are voltage-gated calcium channel from L-type. And then, and then you have in the membrane of the sarcoplasmic reticulum ryanodine receptors. They are also calcium channels, not voltage dependent, but calcium channels. Okay? And now what happens here is this. If you look at the membrane potential, you see here the charge that is inside of that's outside of the cell, that is inside of the cell. Inside is negative. It is resting or action potential. It is resting potential. You see, this is the situation during rest. And now as an action potential is produced, it means the polarity here changes. Inside becomes positive and outside becomes negative. Then this action potential make a conformation change in DHP receptors. And DHP receptors are connected with ryanodine receptor. This conformation change of these receptors changes the conformation of ryanodine receptor. You see the ryanodine was like this and now has this shape. And this change of the shape opens this channel. You see the channel is closed here and now it opens. And the calcium will be released because the calcium inside the sarcoplasmic reticulum is too much compared to the calcium concentration inside of the cell. And now the calcium is released and the muscle contraction can begin. Okay? I want to, even we don't study heart muscle, but I want to mention here what is the difference in this part between the skeletal muscle and the heart muscle. The difference is this. By skeletal muscle, you have both these receptors, but if action potential is moving forwards here, as I told you, that is a voltage-gated calcium channels, that DHP. It opens by depolarization. And now the calcium from outside of the cell can enter the cell as well. And in the skeletal muscle, it is not like this. This is the conformational change which opens the ryanodine receptors, not the calcium entry from outside into the cell. Okay? For example, if you take the muscle fibers and put it in a dish, in a dish which is extracellular fluid, which has no calcium, and now you stimulate the muscle contract. It means the muscle contraction doesn't depend on the calcium concentration outside of the cell. It has its own calcium which is released from sarcoplasmic reticulum. Also, contraction is independent of extracellular calcium concentration, okay? Because this electrical potential or depolarization open through conformation shell, open the ryanodine receptor and release calcium. In the heart muscle, it is different. If you take the heart muscle fiber in a dish which has no calcium and you stimulate the heart muscle, it does not contract. Even the heart muscle has Zarcoplasmic Reticulum and has a lot of calcium in the Sarcoplasmic Reticulum to be released, but that cannot be released if you don't have calcium out of the cell. And what happens with the same receptors? What happens in heart is this. This action potential which depolarizes the membrane opens the DHP receptor. Some calcium comes from the outside into the cell, and this calcium entry opens the ryanodine receptor. And then the calcium for ryanodine receptor will be released. Therefore, it is called calcium-induced calcium release. It means that you must bring some calcium into the cytoplasm from outside through the cell membrane so that those increasing calcium open the ryanodine receptor and release the calcium from inside of the cell. If you don't have calcium out of the cell, then no calcium entering here, and then the ryanodine receptor remains closed, and therefore the muscle, heart muscle, does not contract. So you see these differences between the skeletal and heart muscle. Even both of them have DHP receptors and ryanodine receptor. One is independent from outside calcium, which is the skeletal muscle, and the other one you must have calcium-induced calcium release. Okay, yes, please. Yeah. That is not known exactly, the different structures or not, or I don't know that information, but they have DHP receptors. Okay, now I want to a little bit do the morphology of the actinium-mucin filament. For that part, that I need for explanation of the function, okay? What we have done until now, we had action potential which causes the release of calcium from endoplasmic reticulum. And now we want to see how the mechanical contraction is happening. So we have here the actin filament, which is the thin filament. We have two types of filament in the muscle, thin filament and thick filament. Actin is the thin filament. And you see that is a double strand, composed of monomers of F-actin. Every strand has about 13 monomers. These balls that you see here, they are the monomers. That is one strand, and that is the next strand. So they are twisted with each other in a double helix form. And these actin monomers or filaments have binding site for mucin head. They are these white circles. They are the binding position for mucin head. And normally, these binding sites are not free, not exposed, not free, and they are covered by a protein which we call tropomyosin. That is the tropomyosin. Normally, the tropomyosin lies over these heads. It is shown here differently that you see there is bonding site for mucin. In resting condition, all these binding sites, this tropomyosin is going over the heads like this. All of these are covered by the tropomyosin. Okay? That is one protein which is associated with actin filaments. There is another protein complex, which we call it troponin complex here, associated with actin. And this complex is built of three different type of molecules. One is troponin T. T stands for tropomyosin. You see, physiology is all the name. T is tropomyosin. What does it mean? It means that this troponin binds with tropomyosin. Okay, that's troponin T, binds with tropomyosin. Then you have another molecule which is called troponin C. C is for calcium. This troponin binds calcium, and therefore it's called troponin C. And then you have a troponin I. I is inhibitory. It is an inhibitory troponin which binds with actin. Okay? One of those troponin complex binds with actin, one with tropomyosin, and one binds calcium. And that is the composition of thin filaments. If you look at the thick filament, again you have a double strand here. And you differentiate three different regions in this filament. One is the tail of rod, they call it also rod. And the other one from here to here, that is the hinge. And then you have the head. And you see that here at the hinge region, both these strands floor off and build two Gulliver heads. Okay? And now if you look at the head, and that is important for you to know also, the head has two binding sides. Two binding sides. One side binds with actin. It has a binding site to bind with these positions on actin. Okay? There is another binding site which binds ATP. Okay? And has also the ATPase activity. So that after ATP binds there, it will be hydrolyzed by ATPase activity of the head. Okay? One side to bind with actin, another side to bind ATP. And each head has also two light chains. One light chain red here. It is called alkali light chain. The function of this alkali light chain is to stabilize the head, for stabilization of the head. And there is another light chain here which is called regulatory light chain. If the name says regulatory, it means it regulates something. What does it regulate? It regulates the ATPase activity of the head. Okay, if there will be more activity in breakdown of ATP or less. That is the regulatory function of this regulatory light chain. And also this regulatory light chain, the activity of itself will be regulated. And that regulation occurs through phosphorylation of light chain itself. If it is phosphorylated or not, the activity of light chain itself is different, but then the light chain regulates the activity of the ATPase of the head for breakdown of ATP. Got it? This information. Now we want to see how do they look during rest and what changes we see during muscle contraction. This first picture shows you the situation during rest condition. And you see now that all these binding sites for mucin head is covered by this trophomucin. You see now you don't see the circle very well because trophomucin lies on it. That is the resting condition. And what happens there, that is the trophomucin, which covers here the binding sites for mucin. And now you have troponin T connected with trophomucin, troponin I connected with actin here, and troponin C here, which can bind calcium. You see that this accessibility of this binding site for mucin head is closed. Mucin head wants to go and bind there, it cannot. It has no accessibility, it is blocked. And all what needs to be done here is to make this free so that the mucin head can go and bind with it. And how does it happen? It happens through calcium entry from calcium release from Zarcoplasmic Reticulum. As calcium is released, first I must say this troponin C, which binds calcium, has binding site for two pairs of calcium. One is a high affinity binding site, one pair for two calcium, and the other side is a low affinity calcium binding site. The high affinity binding sites are always occupied with calcium or with magnesium, even during the rest. But the low affinity binding sites also too are free of calcium during rest. And now as action potential comes and release calcium from SR, this calcium binds to the low affinity sites of troponin C. And you see it here, two calcium now binds with troponin C. As the calcium binds, it produces two effects. One is, and you can see why. One is that this troponin E, which is connected to actin, will be dissociated with actin. Because if it is connected, you cannot move anything. And it dissociates the troponin E from actin. And now the whole complex here can move because it is not bound with actin. That was the first effect. And then what it does, this effect is produced through troponin C. Troponin C now roll out towards this trophomucin. Push the trophomucin away. If the trophomucin is away, and this is also dissociated, then the head, the binding sites become available, exposed to the outside, to the other element of the cells. And now the binding sites is open, and the head becomes very happy and say, now I go and bind myself with the actin. Goes there and binds with the actin, and the actin-mucin interaction, we call it cross-bridge cycle. The interaction, the mechanical process of cross-bridge cycling is starting. Okay? Calcium binds with troponin C, dissociate troponin I from actin, move the trophomucin away, and then the mucin head can go and bind with actin, and the contraction, the cross-bridging cycle starts. Now we go to cross-bridge cycle. So that is our starting point. At this starting point, you have calcium, the calcium is not in the cell yet. The calcium is not released. In this condition, the trophomucin sites are all covered by the binding sites are covered by tropomyosin. And if you look at the mucin head, the mucin head already had bind ATP, ATP, and partially hydrolyzed ATP. The hydrolyzation is not complete. It is partially hydrolyzation. You see that ATP is now broke down to ATP and P, but ATP and P are still connected with the head. That condition, I call it partial hydrolyzation, okay? The energy is delivered and the head is energized and want to bind with actin. And now the action potential comes and release calcium into the cell. Calcium is coming into the cell. As calcium enters the cell, then opens the binding sites for mucin, and the mucin binds with actin. As soon as mucin head binds with actin, the conformation of the head changes a little bit, changes. And that conformation change causes the phosphate to be released from the head. And this release of the phosphate causes a conformation of the head, so that normally that is the head, it is like this, in this position, angle of 900, and suddenly it goes to an angle of parting, for example, of 45 degrees. It nicked, the head goes like this. And because it is connected to actin, the actin will be pushed in toward middle of the sarcomere. And you see the head is here going in that direction and pull this actin filament towards middle of the sarcomere, and then like this, if you pull them, it becomes like this. Also, that is the contraction, that is the shortening, what is happening there. And then after it happens, the ADP will be released from the head, okay? And now this head in this position remains connected with the binding site of actin. It remains connected, and if nothing happens, it remains that way forever. To dissociate this head from actin, then an ATP molecule must bind with the head. And what if you have ATP here in the cell, then the ATP goes and binds with the head. As soon as ATP binds with the head dissociates from actin. Also, this dissociation of the head from actin happens with binding of ATP, not hydrolyzation of ATP, only the binding of ATP. Dissociate the head from actin. And you see that is now released, and yet the ATPase activity of the head breaks down the ATP to ADP and phosphate. And that breakdown produces a release energy. And that energy is used to bring the head from this position back to this position. It was like this, now ATP binds and is hydrolyzed, the energy is released, and it becomes like this. The head, the mucin heads, return back to its normal position. And if there is a cell calcium in the cell, then it connects again to another actin. If there is no calcium in the cell, then the contraction stops. It means there are that is called a cross-bridging cycle. This cross-bridging cycle goes on and on and on and on as long as you have in cytoplasma calcium and ATP. These are the two requirements, calcium and ATP. If you take calcium out of the cell, pump it back into the sarcoplasmic reticulum, then the contraction stops. If you have no ATP, the contraction stops as well. When is that the case? If you, by the dead body, after we are dead, all cells, all ATP is not produced anymore. All ATP which were in the cells, they will be used. And now no ATP is not delivered. And therefore, the head remains in this condition connected with actin. Therefore, you see that by dead body, the muscles are stiff and you cannot, for instance, move the legs or the hand after a while. Because the ATP is used, all of ATP, and actin and mucin cannot detach from each other. And it makes the muscle stiff and avoids its movement. That is happening by the dead, okay? So the rest of this lecture will be tomorrow, and now I will go to attendance, okay? I open the attendance for this.